The Dime is sponsored by Eighth Revolution. The cannabis industry has unique challenges, which means you need a multifaceted partner to help you navigate the landscape. Eighth Revolution has a team of experienced science and business experts to provide a unique analytical approach, providing services from capital to cannabinoid and everything in between.
This is The Dime. Dive into the cannabis and hemp industry through trends, insights, predictions, and tangents.
What's up, guys? Welcome back to another episode of The Dime. I'm Brian Fields, and always I've got my right-hand man, Kellen Finney, here with me. And this week we've got a very special guest, Ben Euhus, Cannabis Scientist. Ben, thanks for taking the time. How you doing today?
How's it going, guys? Great to be here.
We're excited to have you. How you doing, Kellen?
Doing good. Really excited to talk to Ben and learn as much as we can about the endocannabinoid system today.
Yeah, I'm looking forward to it. So, Ben, for our listeners who are unfamiliar with you, can you kind of share a little bit about your background?
Yeah, so I'm a biochemist from Texas, trained in Texas State University. And most recently, I finished my graduate program in Israel at the Technion under Dr. Dedi Miri. And we were pretty unique. We had a unfettered access to whole strain cannabis extracts in biological models. We were able to research bioactive compounds and mechanisms of action with cannabis acting on the endocannabinoid system using healthy and diseased models. We were mainly like a preclinical lab that focused on cancer studies. I focused on brain cells that were present in neurodegenerative diseases like multiple sclerosis and Alzheimer's disease. But that is, a cannabis scientist, endocannabinologist trained in Israel. So
cool. Now take us through what your role is currently in the space.
I'm also an ambassador for Dutch Medical Cannabis Society.
Awesome. So let's kind of dive into some of that. I know not every day is the same day. So take us through kind of what goes through, your normal day-to-day process.
So I'm an analytical chemist, and we work on the API formulation and method development and validation to create one of the best possible standards for Texas and hopefully the rest of the globe.
I have a quick question, Ben. You mentioned Dronabinol. Could you kind of walk through what the API in Dronabinol is for our listeners who haven't heard that term used yet?
So Dronabinol is a synthetic THC. It's commonly used for HIV patients, as well as for cancer patients that are experiencing conditions like nausea that are just coming off of chemotherapy. But given that it is a synthetic THC, given that it is a THC-based medication, it does have a prospective slew of beneficial therapeutic potential for conditions outside of HIV and even cancer patients facing nausea.
From like a research standpoint, obviously there's such a misunderstanding across the board of like where we are and where we need to be to get there. So Ben, in your opinion, where do you want to see the research going? Can you talk about some of the recent research you've seen that's really excited you the most?
Yeah. So, when I went to Israel, I was originally looking for a position in science inside of the States that was doing more cannabis-based science. And it kind of became clear that, given the regulation that we have, the restriction, it wasn't really possible to find that. And I was so lucky that I was able to meet Dedi Miri at the Cannabis Science West conference that's held by Josh Krosny and the whole Cannabis Science Conference team. But it became very clear, that this was one of the only places to do cannabis-based research. And originally started with, the love of the potential of these compounds. But I honestly fell in love with the endocannabinoid system, and hoping that as, time goes on and we learn more and more about it, that we are beginning to understand where its stance is in biology and medicine. To me, cannabis is like this key to the endocannabinoid system. It has all these potential activators and agonists and antagonists that work on the CB1 receptors, CB2 receptors, other G protein receptors that are considered orphan receptors right now, but as well as receptors like the trap channels, TRP, which has a whole slew of different constituents. So what we're seeing right now is an expanded, definition of the endocannabinoid system. And the endocannabinoid system is this ubiquitous homeostatic regulatory system that's found in every single living creature. People argue that it's not found in insects, but you can find the trap channels there. So I would argue that it's found in every living creature. This predates cannabis by hundreds of millions of years. There's been really interesting studies that show that aquatic species back, 500 million years ago carry the CB1, CB2 receptors carry the enzymes and the endocannabinoid system's constituents. So we're really kind of seeing how ubiquitous and how prolific this system is. It's found in your circadian rhythm, your pain and pleasure reward systems. It's responsible for the differentiation of what your cells decide to turn into, even programmed to when they die. And really interestingly as well is that on the end of serotonergic, dopaminergic neurons in every single brain that appear and as early as the fetal embryo, you can see the endocannabinoid system, CB1 and CB1 receptors acting as neuromodulators. So it's absolutely essential to most everything in our lives. It is highly deserving of a place in medicine and biology for its potential in therapeutics.
For our listeners out there who are still kind of gathering the initial footing on the endocannabinoid system, in its simplest of form, if the three of us were sharing a joint, would this be why one of us would feel, let's say, sleepy in comparison to the others being more uplifting?
Yeah, I think so. We're, still not even sure as to why, that high or what's driving that, that feeling of the high. The most interesting hypothesis that I've seen and heard is that it's the endocannabinoids that are kind of modulating this. I can give you a really quick example with a compound called oleamide. And this is denoted as an endocannabinoid-like compound. It's not officially in the same realm as like anandamide and 2-AG. It would be plays a role in that entourage effect where it's a steric modulator. But Dr. Meshulam posits that, believes that Dr. Meshulam was the guy that, identified THC for the very first time in 1964, which rippled into us discovering the endocannabinoid system. So very, credible guy. But this compound named oleamide is responsible for a sleepy feeling that we have, a hypnotic feeling. This is also backed up by DiMarzo. So, and, if we're sharing a joint and, one of us feels that it's a sativa and another one feels that it's an indica, it's more than likely that just given our basal levels and what our endocannabinoid levels look like pre-joint and how those are modulated is what's determining how we have an effect. So it's not really, in my mind, as simple as saying that this strain has these terpenes, so that it's a sativa, or this strain has these terpenes, so it's an indica. It's more than likely it's a good guesstimate, but every body is a little bit different.
The many too many problems, like our friend Dr. Abrams usually says, especially when because we talk about, right, like if someone's bringing like a sativa, which we obviously hate discussing that as the frame of reference, but it just for this case point, it's the easiest way for the generalization of this. If you were passing that out and say, hey, Kellen, like here, like you don't need to worry about it kind of putting you in the couch. Here's a sativa. And then Kellen, has to take a quick nap on that. He's like, hey, what's the deal here? And it kind of just puts into mind of how far we still have to come with the understanding from a science standpoint. So Kellen, kind of take it one step further. The ECS, right? Like it's discussed more and more. We're seeing it, but from a doctor's standpoint, I
think we've talked about this previously. The endocannabinoid system isn't even taught in medical school yet. You know what I mean? And so like we do have a long ways to go, but I just want to piggyback on one other thing that Ben was saying in terms of, that's why the need for personalized medicine is such a big deal. You know what I mean? Because like sativa, hybrid indica, we having different base levels of molecules in our bloodstream is going to change how we interact with that product. You know what I mean? And so I just want to drive that point home a little more. But yeah, the endocannabinoid system I've seen a lot of different claims associated with how it like affects our homeostasis, right? You're just like base level of being alive, for those who don't know what homeostasis, what we're referring to. But I've heard it affect everything from mood to hunger to your sleep cycle, everything that Ben was saying. So I think at the basic level, we need more research associated with the endocannabinoid system. And I think that's probably the most fundamental thing that needs to occur in the industry, because like categorization of all these different products and dosing of THC and all these things could just be in vain if we don't understand exactly the mechanism associated with what is getting high. You know what I mean? And we can see that with CBD and THC. You know what I mean? Very, very similar cannabinoids from a structural standpoint. They're the exact molecular weight. One has lactone, one doesn't. Right. And because of that, one small feature, you get a completely different experience than like ingesting CBD versus THC. You know what I mean? And so how those molecules interact with the receptor in CB1. I mean, I haven't read enough primary literature on the crystallography structure of the active site in CB1 and all those things to like truly to be able to disseminate. I haven't seen at least an argument placed before me that can really explicitly define why THC binds so much better than CBD and actually causes that quote unquote high. Have you seen anything like that, Ben?
Nothing as, and I'm not an organic chemist or a structural chemist, unfortunately. So it's a little bit over my head, but
it touches on biochemistry though, right?
Yeah. Receptorology. Yeah. Well, one thing that was really interesting that I reported, and it might have even been in the last decade, was the crazy comparison between THC's tertiary structure and anandamide's tertiary structure. And then a tertiary structure is, the structure that a compound exhibits when it's binding to a receptor. This lock and key kind of method for biology to activate different receptors. But when you look at a picture between anandamide and THC, the similarity is just ridiculously, they're almost identical. So I'm not really sure what's going on because, if you were to, take them apart and put them in their primary structure, like, if you're just to draw them on a piece of paper, they look like very different compounds. So I'm not really sure what's going on in that respect. There is, some things going on inside those receptors that would be very interesting to see.
What is anandamide?
Anandamide, sorry. So anandamide is an endocannabinoid that is, the acronym is AEA. And anandamide was the first endocannabinoid discovered, and it's discovered by Meshulam, I believe, or another host of scientists, and was given the name anandamide, which is Sanskrit for bliss molecule. And so this was the first ligand that they found to bind to CB1, and, back in a time when weed wasn't 30% THC and was more likely to make you very blissful, I guess they felt that it was a very suitable name.
Thanks for breaking that down. So let's I can spend the entire episode asking why we didn't learn about it in college, why the doctors aren't taught this.
Right.
That's the past. Let's talk about the future. Personalized medicine. What sort of roadmap would you like to see in order to take necessary steps to get there?
Yeah. So even outside of the field of cannabis, to look at to see, like ketosis, for example, a lot of people are doing these ketosis diets. And that's, essentially when you starve yourself of carbohydrates so that you're producing these ketone constituents inside your brain. Inside of that realm of even just dietitians, it's extremely, extremely difficult to get a proper analysis of what's actually going through your body right now. Outside of a spinal tap, even, it's very, it might be even impossible, but it's very difficult to determine what's responding in your brain, what's being produced in your central nervous system in your brain. So a roadmap would have a higher level of identification for, these endocannabinoids that are circulating through your body. We could have a better way of understanding what's actually being activated and upregulated. Right now we're using mice models, and these mice are being sacrificed, and we're able to see a differential ability for the production of endocannabinoids. One of our studies, or a few of our studies from Israel, from the Miri lab, really worked on looking at the difference in endocannabinoid levels when given, strains with almost identical THC, CBD levels. And we're seeing that there's a tremendous difference in the endocannabinoids that are being produced strain to strain, even if they have the same THC, CBD. So something that would be really important for us is to see where a patient's basal levels are. And even in the recreational, recreational consideration, where a person's basal levels of endocannabinoids are, and how they're modulated after, inducing some kind of cannabis product or, cannabinoid-like product. And another thing that is extremely important is refining our analytical techniques. We're so lucky that we are where we're at because, 40 years ago, we, didn't have things like HPLC or LCMS. And this is, one of the reasons that they weren't really definitively able to show how important, cannabis or what inside of cannabis was actually producing a therapeutic effect. But right now we're able to see that there's, 96 plus, 100 plus cannabinoids in any given type of strain. In 2018, or even as far as, most recently as 2020, when I left my graduate program, there were still like 17 cannabinoids that were unidentified, and they had names like 31317B and things like that. And they seem to have a role in the bioactive effect. So what we need right now is, a way to determine endocannabinoid modulation and a more refined way of determining what's actually inside these strains.
Do you think we'll get there in our lifetime to personalized medicine?
I think so. I think the field of personalized medicine is just exploding right now. One of the things that's really kept or put cannabis science into semi-warp speed is the amount of information that comes from outside disciplines. So we're using techniques and, even recruiting people from outside disciplines, interdisciplinary fields that are, just constantly pushing, the boundary of what's possible. So yeah, the field of personalized medicine, I feel like, is just, refining, and there is, so much interest and, financial incentive to actually, pursue R&D. I think it would be a matter of time for something potential inside of analytics to help us answer these questions inside of cannabis. So I'm pretty optimistic about it.
In terms of the personalized medicine, I think that one of the biggest obstacles is, like you were saying, being able to measure exactly what's going on inside the brain, right? Because we can talk about those cannabinoids and you're smoking them to go into your bloodstream, but like what truly goes through the blood-brain barrier? You know what I mean? A little shout out again, I'm going to try it, another Hail Mary here, but Elon Musk, right? And Neuralink, by actually implanting a chip though, and if you can put some like small sensors in there and like being able to run some chemometrics and like be able to measure cannabinoids, like this could be a huge opportunity for Musk to make some money.
Yeah. This is like the cross-disciplinary, outside disciplinary field. And
but that's how you could do it. You know what I mean? Right?
Yeah, I think there could be something there. I don't know how far along they are with the Neuralink, but I guess they're putting it into chimpanzees at this point. So I guess
they're playing video games with it already.
Yeah. I just want to go back to what Kellen said, and he said there's a chance for Elon to make some money. Elon doesn't solve problems to make money. He solves problems that he thinks are necessary for like human survival, right? Like that's where he's at now, not trying to make money. But to continue on that path, maybe Ben can come back at a later point and update us on the comparison on the Neuralink and the endocannabinoid system. Let's dive into your research in your graduate program. I want to learn more about that. We heard your talk, roughly 2 years ago. It was incredible. And I want you to expand on some of the areas from a medical standpoint that you kind of came across or the research that fascinated you.
Awesome. Yeah, absolutely. And, I wrote a small publication for Cayman Chemical, which is a pretty succinct, missing some, details and major points. But it was a succinct kind of explanation of big patterns that we found. And the whole thesis essentially was cannabis is a great key to the endocannabinoid system. But inside the lab that I was with for 2 years, we were a preclinical, largely anti-cancer or cancer therapeutics, chronic disease-based lab. So a lot of my colleagues were working on cancer projects, and I was working on a project with microglia. And microglia is the primary cell inside the central nervous system and brain. It's kind of like the primary sentinel that's looking around, repairing things, eating up stuff, and alerting other types of immune-based cells to drive an effect. But some of the biggest things that we found were that cannabis has such a differential ability strain to strain. And one of the things that really drives this was a colleague's, it's in the published paper. There's a few studies that I'll bring up right now. And one of them was a colleague's study that did they had 12 different cancer cell types. And one thing that they found is that not every strain has the same ability. Even if they are in very comparable levels of THC and CBD, it's not given that they're going to have the tremendous ability. One thing that was also found in that, or two more things that were also found in that paper is that not every single cancer cell type is susceptible to cannabis treatment. So, in most cases, there were two different types of cell of the same cell. So, you can, when you're doing cell biology, you can get a few different cell lines that are indicative of a type of disease. So, we had 2 different breast cancer lines, 2 different colon cancer lines, et cetera, et cetera. And in some cases, not, both breast cancer cell lines were not affected, or not, both colon cancer cells were not affected. And sometimes you saw like just a tremendous differential ability, where the breast cancer was extremely susceptible. But cells like HT29, which is a colon cancer cell, had almost no susceptibility to being treated by cancer by cannabis. And when I say susceptible, like literally what we were doing was admitting a tiny amount of cannabis extract and tracking to see over the course of 24 hours how many cells died. So literally, it was walking into a lab that was killing cancer every day, and that's just it was pretty cool in itself. But another thing that we found, and or another thing that was found in that study, and there's such a difference in from higher levels of complexity in organisms. So it would be interesting to see if this is something that also carries through in people. But the carboxylated forms of THC and CBD, THCA, CBDA, had a terrible ability at inducing cell death in cancer cells. So, your formulation also plays a big role in, the way that a, cannabis strain or an extract is going to, affect a certain scenario. So, one more thing that I wanted to share in that same realm was a study done using mice. And what we did was induce an epileptic response using a compound called PTZ. And this is a really common thing to do when you're looking at epilepsy and therapy for seizures. But we used 5 different strains, and looking at the response with the mice, what we looked at was the time in between seizures, the overall survivability, and how they fared basically. So we used 5 different strains, and one thing that was seen is that the survivability went up for all the mice, regardless of the strain that you use. So that's very positive information, I feel like. But one thing that we saw was such a difference in the latency, which is the time it takes for them to have a seizure. So there's a tremendous difference ability in therapeutic potential just between these 5 different strains. And the kicker was that each strain had exactly the same amount of THC and CBD. So THC, CBD is a terrible indicator for determining a potential therapeutic response. It might be better to, as a guide versus looking at like a strain that's high in CBG or high in CBD, what have you, or THCV, things like that. But we're shooting ourselves in the foot by just using those 2 compounds as an indicator for therapeutic potential.
Yeah, that's exactly what happened to GW Pharmaceutical too, is they had to literally grow the exact same strain indoors in order to be able to produce the exact same chemical profile of Epidiolex.
Right.
Is literally that exact issue, is they discovered that it wasn't the ratio of CBD and THC, it's everything else that comes with it from an extraction standpoint.
And so one of the, the things that I kind of harped on when I was there, it's not really the levels that we should be concerned with. Maybe it's an indicator that, strains that are high in CBD are carrying other kind of beneficial types of compounds, but, and maybe they're acting as the vehicle, but it's more of an indication, I think, that something is inside of it that is acting or helping drive the effect. And I just want to touch base as far as the carboxylated compounds. While they were not very helpful in the cancer study that we did, in my research dealing with microglia, and one of the things that we looked at was how well these cells were able to respond to wound injury. So essentially, if you had some kind of neural damage, which is what's happening with a disease like multiple sclerosis, we wanted to see how fast and how much better these microglia were able to respond and make some kind of repair. In this scenario, the carboxylated strains were tremendous, and they were the ones that we followed suit with. So it just entirely depends on the situation, entirely depends on the condition that you're looking for and the outcome. But it does seem to have, both parts, both forms of the plant, seem to have, tremendous effects in their own respect.
So there's a ton to unpack. So I have to take a couple steps back, and I apologize if I kind of misspeak. So when you were doing the cancer studies, right, you had identified specific strains worked better for specific types of cancer. Is that how I
Yeah. So certain cancer cells, cancer cell types were more likely to be affected by just cannabis in general. And then certain strains were more likely to induce a positive response. And one of the
But
things,
not CBD and THC as the main component.
exactly, something that that's found in this, same publication. And I think it's, if anybody wants to look it up, it's Baram 2018 or 19.
We'll include a link in the show notes.
Shout out to all listeners who check that out.
Yeah, it's the paper's title is a mouthful, but one thing that they did is, they saw using about 16 different cancer cell types, they saw that some strains had a more consistent ability to induce cell death. And what they did is take one of these strains, which was high in THC, and they performed, took a gradient of different strains at different levels of THC. So we had, let's just say like 12, it might be something like this. So don't quote me, but it might be like 12 different strains or so, ranging from 1 milligram to, 4 milligrams of THC, per percentage of, the entire plant. And they also used a purified version of THC. And what they did using these different strains at variable levels of THC was, determine how well they were at inducing cell death. And very surprisingly, the purified THC didn't seem to have any effect on inducing cell death at that level. And there was absolutely no way to determine a strain's ability to induce cell death based on its percentage of THC. So the highest performer was like 3.6, 3.2, something around there. But the strains to the left and the right of it did not indicate that this was, a gradient or anything that they had shared in common outside of their THC levels.
Let's take another step forward in this direction, right? It's a promising step. What's the follow-up after a trial like this? Is it additional trials in this direction? Like, take us through kind of how that works.
So one of the things that, really is essential to transform this into a clinical trial is determining the bioactive compounds. So one of the things that we did was a process called fractionation. And fractionation is a pretty old technique. It's pretty used in the field of just in biomedics in general. It's used outside of cannabis. It's used in other Israeli labs that are able to access this research. But essentially what they do is use a bit of column chromatography and kind of partition out based on time and size of compounds, and partition out the compounds in a cannabis extract into different little components. And you do this so much that the idea is that you're getting the most isolated versions of, compounds present as possible. And so what you do is, you make about like 5 to 7 different components. The first one's got THC, the second one's got CBD, the third one's got, THCV, or the whole, slew of minor cannabinoids. And you go through and just, determine at what combinations or, if there's any combinations that are working. And you just kind of repeat this process until you're left with the most isolated versions of these compounds and can kind of determine what's going on. And just in the whole aspect of our refining of analytics, this is a process that is very difficult to get a good source of these components, because these compounds are coming in at such a small amount of the extract that it's very, very difficult to elute them out at large enough levels. Because after you elute them out into different components, you have to go through a process of refining and purifying them. And through any step along the way, you're going to lose some product. But essentially in research, looking at the bioactive compounds in cannabis kind of follows this suit where you're kind of eluting them out. And that could use like a lot of revision. But, as far as Epidiolex and even, Bedrocan, the Dutch medical cannabis program, they have a very strong backing on their genetics that they're using. They want to have the absolute most consistent plant time after time after time. Because one of the biggest pushbacks that we've seen in the field of pharmaceuticals or clinical science using cannabis is that it's extremely difficult to have a consistency in botanical formulations. So even if we were to see an extract that was very, very helpful, in the lab, if they're not able to replicate it, time generation after generation, and this happens too. This happens in Israel. You can have a very therapeutic response to it one generation, and then the next generation it doesn't work, or even worse, it brings about a serious adverse effect. So those are things that they do. They are still trying to just make a consistency in the genetics and find strains that are helpful to patients, and just pray that they can replicate those that plant composition. And they're also working to determine bioactive compounds through processes of elution.
Well, this kind of might even bring it full circle back to synthetic chemistry as potentially the answer. This is what they did for Taxol, right? The chemical from a tree that's a cancer drug, right?
My favorite example. So, as this comes from the Pacific Northwest, this tree, the Mountain Yew, and it's a chemotherapy drug that's was determined to be extremely effective, and it got to a point where there wasn't enough trees to even produce the level of demand for this this medicine. So very smart people started synthetically producing Taxol, and it's, at least in the very beginning of the 2000s, it was the highest selling chemotherapy drug, if not one of the highest selling drugs on the market. So there's a high demand for it. It's very efficacious. And it's the synthetic chemistry that is, really making this a possibility for the modern world.
Ben, your opinion, do you think pharmaceutical companies will help the cause for cannabis as a therapeutic, or do you think that they're look to push against it?
Honestly, I'm pretty optimistic. I believe in the cooperation between pharmaceuticals and cannabis. I don't have the stance that pharmaceutical giants are out to destroy us. That argument can definitely be made, and I don't know if we have enough time for that entire one. But just speaking to doctors and physicians that are trying to get access to Epidiolex for research studies, or even through patient access, one of the biggest things that they've said time and time again is that the FDA, DEA are very cooperative, but they've boxed themselves in. There is this extreme regulatory framework that the FDA, DEA has set up against themselves that has boxed them in. So they don't have the ability to want to just drop it but they don't have the regulatory ability to engage in this activity just yet. But it does seem like they do like to cooperate, within reason for drugs that have passed the clinical pipeline that have been proven to be effective. So I'm very pro pharma. And I think that, there are very positive things that will come out down the line with it. But
Kellen, do you want to take your natural stance of anti-pharma?
No, I think that from an optimistic perspective, the biggest challenge is going to be we've literally been discussing how specific strains have different effect. And I think to kind of distill that down to its essence is different strains are going to have different chemical profiles, right? And at the end of the day, we're seeing these very complex extracts that could have upwards of 400 chemicals present at one time, right? Just you're manipulating a few of these different chemicals in very, very small concentrations, right? Because I don't know exactly what the potency of cannabinoids in terms of mainly CBD and THC were in your extracts, but I imagine they're above 50 to 60% in the extracts. Right. So we're talking the manipulation of the other 40%, and with 400 chemicals over that 40%, we're talking potentially a couple milligram difference in an active chemical. And it's just it sounds like a very steep hill to climb to go through and identify exactly which chemical at which concentration is the one that's responsible for what we're seeing from a result standpoint in how effective these drugs are at that point, right?
And a lot of the tools that we're using in this research, are, because of the pharmaceutical industry.
Yes.
I understand that the DEA kind of got us into this situation at the beginning of the century, or beginning of the 20th century, sorry. And if we have the analytical capacity that we do now, back then, there was cannabis appeared in the American Pharmacopoeia as late as 1952, I believe. And there were physicians and pharmacists that were really advocating that the therapeutic potential of this plant has not yet been fully elucidated. I think if we had that capacity back then, there's no way that it would be the same situation that we're in now. So, I do realize that, DEA has put us into this, and there's just been a, crazy amount of repercussions extending, not even just in medicine, but, in civil rights that, are because of the, the regulatory framework, or even how they're planning or how they deal with it. But I think the times are changing. And I think that, people that are inside those, regulatory groups right now would be hard pressed to say that it does not have, therapeutic potential. And, just in terms of descheduling of CBD, the descheduling of THC, these are baby steps in the right direction, but I feel like indicative that there is a cooperation on the horizon.
Yeah, I think that as daunting as that task could be to figure that out, if you put the fundamental research on the shoulders of universities across the globe, I think that they'll be able to kind of push through with that kind of an act.
I can't wait, honestly. Yeah.
I feel like we're about to go through like a period of time where there's going to be just massive breakthroughs, and it's just going to be so exciting to see like all the progress we're making.
Yeah. And, even outside of, because there's like little blips of this. And one of the most fascinating endocannabinologist scientists to me is this, I think her last name is Mueller. She's a German scientist who's treating Tourette's. She's got this conjugated system using an antibody-based delivery tied to PEA, which is a endocannabinoid. So I think that we'll, in the realm of medicine and drugs, begin to see compounds like that. We'll start merging cannabis compounds with, monoclonal antibodies, bispecific antibodies, things like that, inducing it in adjuvant therapy with chemotherapy. And I think that we'll just see a tremendous benefit that otherwise wouldn't be possible using these compounds by themselves. So I'm very optimistic about it. They need to teach this in school for sure. But, I do think it's on the horizon.
One problem at a time. So question for you, Ben. What is one area that the everyday consumer of cannabis might not be aware of just from your understanding or research, something that you've come across that you say that most people would be surprised or be shocked by this?
I mean, the field of the endocannabinoid system in general, or what we know about the endocannabinoid system in general. I tell people all the time that cannabis co-evolved with the ECS. Cannabis led us to the endocannabinoid system in the lab, but in nature, the endocannabinoid system led to cannabis. This is a series of receptors and enzymes and ligands found in all species that predates cannabis, that predates trees, that predates flowering plants by hundreds of millions of years. So when I'm talking to most cannabis users that are familiar with CB1, CB2, the extent of how prolific and ancient this system doesn't seem to be understood or common knowledge just yet, which is extremely frustrating. So, and I just feel like on a common level, what we know about the endocannabinoid system really needs to be identified and just grounded in to our cultural lexicon.
Since you've been in the cannabinoid industry, what has been your biggest misconception?
Biggest misconception, I guess that, that THC and CBD are the only compounds. But that's not the same problem that you find with cannabis users. At this point, they kind of know about, CBG and CBN and things like that. But I guess that, the anti-cancer properties lay, exclusively in THC and CBD. But even using CBG, we've seen at least in cell research that it produces a tremendous effect.
What's your favorite minor cannabinoid that's not really getting the most publicity out there?
Do you think is CBN, is that considered a minor cannabinoid?
Yeah, but that one's getting a lot of love. Let's go a little deeper.
Okay. I don't know. 313-16B. Yeah, I would say that, honestly. They're one of the compounds that is unidentified. There are glimpses in, cell and in vivo research that shows that this might play a role in the bioactive effects that's emitted from these extracts. So I'd have to say one of the ones that isn't, fully identified yet, because when we're doing these fractionation methods, it seems that those compounds are actually playing a significant role, which just blows my mind.
Can I find that on the internet?
I will have to take a look, but I'm not sure yet.
Can I Google that? What was it? Just Google that exact.
What was it? 313?
Yeah, 313. Yeah, I shouldn't even be talking about it, but you can look.
People are going to show up at Ben's house now, knock on the door, take him away.
My old PI is going to show up in my house and take me away. So the IDF is going to show up. But if you look at publications from the MIRI lab, one of the things that you can see between the difference in the PTZ study that I mentioned with the mice that were induced with an epileptic response, and even the paper that they just released in February of this year that was actually using patient samples and surveys, is this tremendous difference in those unidentified compounds. So it remains to be, seen which one of those could carry a bioactive compound, but it does. We just don't know as much as we think we do at this point.
That's so dope. If you could sum up your experience in the cannabinoid space into a main takeaway or lesson learned to pass on to the next generation, what would it be?
One thing that it seems that what really is essential right now is an understanding of analytical chemistry, bioinformatics, in silico, which is like computational, like ligand binding and stuff like that. So I really think that like what is going to be most helpful to this field is this interdisciplinary cross-discipline expertise that is so helpful in the field of medicine right now. And that seems to be like where even, the recreational and the medicinal space are most lacking, is with analytical chemists, bioinformatics, computational scientists, things of that nature. And it's more than likely that is going to be what really helps us elucidate what the heck's going on with this plant.
Prediction time. 5 years from now, it's 2026. Have we achieved a breakthrough in cannabinoid research that has changed the space medically forever?
Man, I don't know. Where were we 5 years ago? Like, let's think. In 2016, I guess Epidiolex had been released.
It hadn't been rescheduled though.
Okay, right on. And it wasn't even, out in a couple countries. And, even a few states hadn't even, began the process of the medicinal market. So I don't know. It's hard to say. There has been tremendous change in the last 5 years. There's been tremendous change in the last 10 years. But seriously, like what we know about the endocannabinoid system and everything that I've kind of said in regards to that was discovered before 2012. So it's out there. It just needs to be brought to light, in my opinion. So yeah, I don't know. Baby steps, to be honest. And I guess it depends who you're asking. But since you're asking me, I would say that these would be tremendous steps. Like I'm so gung-ho about just the minor incremental steps that we're taking. And even if, because I'm in Texas right now, so even if Texas was just to offer an expanded list of conditions offered to the medical marijuana program, I think that would be just a tremendously large step. So
you and the other Texans we talked to.
I'd say that. Yeah. We need you on the record, though, Ben. Have we achieved a big breakthrough by 2026?
I'm gonna say shit. I'm gonna say maybe.
Maybe.
It depends on the access. Israel is doing an incredible job, and it's they're one of the only countries right now that's using the access they have. I'm not really sure what's going on in Canada. I never worked there before, but they do not engage in the same biological experiments. DeMarzo is like the man in my book, and he's just done so much for this field. It's most people can't even hold a candle to him. But unless countries are able to increase their access, more than likely we're going to be somewhat in the same space we are now. But I do want to harp that even though we don't have like biological access in the States, the recreational industry has really driven a lot of innovation. Without the recreational industry, we would not see this, anxiolytic effect of CBG, these anti-anxiety effects of CBG, the, use of CBN for sleep and things like that. So I do believe that the recreational, industry drives innovation. But in my mind, there is not another country like Israel that's producing the same level of output necessary to really get us to a point where we have a fine holding on everything this plant has to offer.
So I'll have you down as a maybe leaning yes.
Yeah, there we go. I like that.
Kellen,
I say yes. And I think the breakthrough is that we reschedule THC and CBD and you get access globally. Universities get access to it, which I think then will be kind of like the catalyst for just a waterfall of innovation and deeper understanding associated with the endogenous chemicals in the plant across the board. What about you, Brian? What's your prediction? Let's hear it.
So usually I like to take the opposite of the consensus, but for this one, I agree. I think it's just going to be like a stepping stone of advanced innovation, and it's going to be fast. I think there's going to be breakthroughs left and right because we're finally just going to open up the gates and say, all right, guys, like have at it. Like, let's see what we can find out. And even from the early studies, it seems like it's super, super promising. So having advanced opportunities to kind of expand on this would really be promising.
Yeah. And I'll just say one more thing. You can see what types of clinical studies are being undergone right now. You just go on like clinical.gov, I believe it is. But if you Google that, you'll be able to find it. About 2 years ago, and don't quote me this, but like a few years ago, there was just a tiny amount of clinical trials undergone using cannabis compounds that were being undergone. Is that the word? But right now, I believe that there's, upwards of over 70 or so just in the States alone. And they vary between, can you drive on this to the effects of, people with multiple sclerosis. But there is a tremendous amount of clinical trials that were not being, the list has just grown every single year. So that to me makes me feel very optimistic.
Yeah, just the idea that like cannabis can help people medically, I think is like kind of sometimes like the silent story in the room with all of the noise that's going on. And I think it's so promising that at the end of the day, if this product can help people, we should allow others to kind of benefit from it. And if it helps people, that's all that matters.
Yeah, cheers to that.
So Ben, for our followers who want to get in touch, they want to learn more and they want to pick your brain about this new cannabinoid, where can they reach out to you?
So you can find me through Viridian, and we spell that VERIDIAN. So you can find me at ben.viridian@gmail.com. Viridian is the color green. VER is the Latin root for truth. So we are green truth, basically. You can also just feel free to reach out to me at benoyhus91 at gmail.com. Welcome any kind of collaboration question or anything of the sort.
Thanks so much for your time, Ben.
Appreciate it, guys. Pleasure to be here.